We will start a new topic. Before that, from the previous class, whatever the question we discussed, do you have any problem? Uh, if you have, you can ask now. Anyone? No. Okay, then we will move to the next. Okay, today we are starting that from the cell biology. All right, cell biology. Of course, I am in the cell biology. I am not starting first the cell structures, but that should be the first one. But today I am not starting that one. We are going to the more important one is the cell division. All right, cell division, and in the cell division we will start with that the mitosis. Cell division, the uh, mitosis we are starting. Okay, before that, see here that all the cells, now what we know that the growth and reproduction, these two are very important characteristics of all living organism, is it? All living organism grow and they reproduce, they forms their copy. Hmm. That is the reproduction. They produce the similar organism. So, and we know the cells are the basic structure of a structural and functional unit. Cells are a structural and functional unit, is it? So, a functional unit of all living organism. So the cells are actually their structure and all the function at the cellular level. So the cells also cells grow and they reproduce also, but they divide. And we know in the cell theory, you remember that cell theory? In the cell theory, two things were very important, that all the organisms are made of cell and the cell product. And another one, that new cell arise from pre-existing cell, existing cells. From the pre-existing cell, this new cell arise from pre-existing cell. How? By their division. By division. So new cell arise by the division of the pre-existing cell. So that we will learn today, and there are two basic type of the cell divisions are there. As we know, one is mitosis and another one is meiosis. Out of that, mitosis is very common. It takes place in all the somatic cells. Somatic cells means body cells, and meiosis, that is attached with the reproduction of the organism. Reproduction of organism. So this takes place in the reproductive organ, right? So this another type of course, cell division is there. Sometime we call amitosis. Amitosis. Sometimes some of the very primitive organism, unicellular organism, there is amitosis in which this is here not this true mitosis division. Mitos actually means thread. Okay, so thread. So this is that the thread means here is the chromosome. Thread is here the chromosome. So in the mitosis, there is no nuclear events or this chromosomal events are seen. So those are in like sometime in case of amoeba, in case of some bacteria, this division where we cannot see the nuclear activity, nuclear event. So they are called the amitosis type of division. Otherwise, mitosis takes place in all sort of organism. Here some of the points that are related to the that all organisms start their life from a single cell. So when it is starting from single cell, from the single cell, multicellular organisms that develop. So number of cell increase by the repeated division of that cell. Here is that a cell that divide, no? Cell divide, and after the division, then again it grow and divide, grow and divide, grow and divide. So this the cell division, this process, this goes like a cyclic way, and this is known as cell cycle. Okay, and in the cell cycle there is there is two phases are there. One is interphase and one is M phase or metaphase, mitotic phase. Actual division and actual actual division is there. So there is cell cycle. In the cell cycle, there is a sequence of event is there. In this sequence of event, that the cell prepare for the division, and after that we can see some division of the nucleus, and then ultimately the cell divide into two. Then from one cell two cells are produced. After the two cell produce, they are very young, small, and they grow. So this they grow, attain maturity, again divide, attain maturity, again divide. So there is two successive. One is that when the cell is dividing, and another one is time that is passed in between the two successive division. So in the cell cycle, two phases are there. One is called the interphase, and another is the M phase. M phase, or this is the mitotic phase. Mitotic phase, or this is actual division. Okay, and the interphase is in between two. So here you can see in this diagrammatic representation that this Here in this is a diagrammatic representation which is showing that cell division. See here, this blue ring that you can see, this is the interface. Okay, interface, and this one, this portion, it is showing that M phase. This is actual division. What we can see that this interface, no, this is occupying the bigger portion. About 95% of the is total time of the cell cycle is covered in the interface, and this M phase, divisional phase, means it account only 5% of the time. Say like in human beings, where there is 24 hours, a cell cycle is about of 24 hours, but this division is only 1 hour. Mitosis is only 1 hour, and 23 hours is that interface. During this interface, the cell prepare for the division. Here is the cell, actual division of the cell, and this is the phase where it prepared. So initial stage, when the two cell has been produced, that first stage is the G1. Okay, G stand for gap. This is a gap between the M phase and division and the S phase. S stand for synthesis. Synthesis of what? Synthesis of DNA. So in the initial G1 phase, cell, young cell, it grow, it is very active, and it will grow, it will synthesize its material, protoplasm, and it will attain maturity. So cell is very active at this phase. After that, if the cell is going to divide again, this cell is going to divide, then it will enter into the S phase. If the cell is not going to divide further, then it will come out from here, and then this will get mature, get differentiated, and carry out other function. And this is so, it is known as G0 stage. This is here, it is showing G0 stage. At the G0 stage, cell do not divide. Or you can say those cells which are not dividing again, they come out of the cell cycle at the G1, after G1, and they are in the G0. But if the cell is going to divide, then they will prepare themselves for the next division. And for the preparing themselves for the next division is required that the genetic material, the DNA, that genetic material, the DNA should multiply, and it should become double. All right. So here in the S phase, DNA synthesis takes place. DNA as well as histone, right? Histone protein, which is the part of the chromatin. So this is synthesized. This is S phase. So whatever the DNA is there here in G1 phase, by the end of the S phase, that amount of DNA increase to double. After that, it comes to It is the G2 phase. Here, all other protein, RNA, etc., this protein, RNA, and other substances, they are also synthesized for the or those things which are required for the cell division, they are synthesized. Okay, then next start the after the G2 phase, start the M phase or the metaphase. This M phase has the stages. M phase has divisional stages. These are M or mitosis, mitotic phase. This has several again sub stages. They are prophase, metaphase, anaphase, and telophase. Okay, so there is M phase here. It divide. Now, what happened that the DNA, you remember that after the cell division, when from in the mitosis, one cell divide and give rise to two daughter cells. And these two daughter cells will have same amount of DNA because genetic material must be all all the genetic material should come to this daughter cell. They will have the same amount of DNA in these two, means equal amount of DNA as it was in the mother cell. Okay, and the number of chromosomes also will be same. We know that the DNA determine the character of an organism and DNA makes the chromosome. So during this cell division, the chromosome must be distributed equally to the daughter cell so that the DNA is also distributed equally. And here what happened that that's why the two cells, daughter cells which are produced, these daughter cells are identical. Identical by quantity and quality. Okay, quantity and quality should be equal. And as they get the same or equal amount of the genetic material in the daughter cell, this division, mitosis is also called equational division. Well, division. Mitosis is equational division because among the daughter cell, that is the property of the mother is distributed equally and of course in the same amount, and that is why say if in the mother cell there are 16 chromosomes, daughter cells also will get 16 chromosomes. So what is required that before dividing, the mother cell should increase the chromosome number or the amount of DNA should increase. Actually, in a like a number of chromosome we indicate by n or by x, is it? In the similar way, that amount of DNA is indicated by C. In a haploid cell, haploid cell, the number of chromosome is 1n, and content of DNA is 1C. In a diploid cell, in a diploid cell, number of chromosome is 2n and amount of DNA is 2C. So, say one cell which will divide before division, if a diploid cell is dividing, the before division it has 2C amount of DNA and 1n number of chromosome. During this process of preparation, this DNA is synthesized and amount of DNA become double. So it increased to 4C. But what happened? The sorry, here it is 2C. So in the diploid cell, chromosome also will be 2n. Here the chromosome number will remain the same, but the amount of DNA will become double. So amount of DNA become double. Double when it become? It become double during the S phase. During this S phase, DNA is synthesized, and whatever was the amount of DNA in the G1 phase, here now the amount of DNA is double. Here it was 2C, here it will be 4C. In the G2 also it will be 4C. And in the some of the phases of the cell division, it will remain 2C, but after cytokinesis and telophase, it will become again 2C. From 4C, it will become 2C. So this way it continue that the amount of DNA increase to double, and after the division it come back to its normal condition. In the plant, the mitosis takes place in both haploid as well as a diploid cell. Both can divide by mitosis. But in case of animal, mitosis usually takes place in the diploid cell. This is because in animal diploid, leaving some of the very that primitive animals, they in case of plant, both haploid and diploid cell can divide by mitosis. Okay, let us to the uh, come to this mitotic cell division. Here you can see our that what happened in the interphase G1. This is the G1 phase, S phase, F animal cell. Okay, one thing is that in case of animal cell, the centriole also duplicate at the S phase. This centriole is absent in plant cell. In animal cell, centriole is present. So during S phase, when DNA is replicating, the centriole also replicate and become double. G2 phase cell, those cells which do not divide, they are in the quiescent phase G0 of the cell cycle. Then come to the mitosis. This mitosis has several phases. Here prophase, metaphase, anaphase and telophase. In prophase what happened that prophase stage, prophase begin with the initiation of the condensation of chromosomal material. Chromosomal material means that is the chromatin. Okay, this in prophase there is progressive initiation of condensation of the chromosomal material. So chromosomal material when it's start condensing, then what happened that very thin thread like structure chromosome start appearing. Okay, and as the time, as the prophase progress, the chromosome become more and more distinct. Chromosome become short and thick. So under microscope we can see them in a better way. All right. Here is that it is showing the stage, that is the prophase. I'll take it here. Can you say here this is the early prophase. Actually, in the early prophase, the nucleus is very big. It is surrounded by a nuclear membrane, and this chromosome, the thread-like structure, are seen visible within the nucleoplasm. They were not very clear, but as the process progress, we can see within the within this we can see very long thread which are very clear, and as gradually it further progress, this there is thread like a structure, definite structure are seen, and these are the chromosome. In each of the chromosome we can see that there are two threads, and these two threads are joined together at certain point. This portion is the centromere, and these are called chromatid. Okay, so two chromatids in each. Here you can see there are two chromatids, and these two chromatids, they are at certain places joined with each other, centromere. As the time pass, we can see that the chromosome are more distinct, but they are short, means it become shorter and thicker. So they are clear, more and more clear. But during this process, what happened? This nuclear membrane start disorganizing, and by the end of the prophase, all the nuclear membrane is vanish. Okay, so this is what happened, and here you can see prophase begins with initiation of condensation of the chromosome. Chromosome become progressively thick, short, and more prominent. In each chromosome, there are two chromatid cells at centromere. In the animal cell, centriole start moving to the opposite pole. This is the animal cell. And towards the end of prophase, what happened? A spindle fiber, thread-like structure start forming, start forming in between the two pole of the cell. And nucleolus, say within the nucleus, there are a structure called the nucleolus. This nucleolus also disappear, and nuclear membrane disappear. With the with the disappearance of nuclear membrane, the nucleolus, the formation of the spindle fiber that signify, that indicate the ends of prophase, and where the prophase ends, that start the metaphase, that is the middle of the phase. This phase is very short period. This is for a short period, and in this phase what happened, that when it is metaphase, means there is no nuclear membrane, no nucleolus. This centrioles in animal cells, so they have moved to the opposite pole. They move to the opposite pole, and in between the centriole, or in between the two pole, spindle fibers, the thread-like structure has developed, and the chromosome which are more prominent now, but they are shorter and they are thicker. These chromosomes are attached to some of the spindle fiber. Okay, some of the spindle fiber, the chromosomes are attached. And what happened in the prophase, the chromosomes are actually scattered in the cytoplasm, because the nuclear membrane has lost. So now it is, there is a nucleoplasm and cytoplasm, they are continuous. So they have scattered, they were scattered. But as the metaphase start, the gradually the chromosomes they align themselves in the center of the cell. That is the equatorial plane. Chromosomes are aligned in the equatorial plane and the chromosomes are attached to the spindle fiber. Say chromosomes are more thick and short. Chromosomes have aligned themselves with their centromere along the cell equator and attached to a spindle fiber. A spindle fiber, there is uh, these spindle fibers are actually attached to the kinetochore. Actually, when there is, say, this is this one is the chromosome having two chromatids, and at the chromosome, this is centromere where the chromosomes are attached, chromatids are attached together. At this place, another disc-like structure is there, and this is known as kinetochore. Kinetochore, this is a disc-like structure present on the surface of the centromere. Okay, and to this kinetochore, the spindle fibers are attached. This is a spindle fiber attached to the kinetochore. Okay, the plane of alignment of the chromosome at metaphase is called the metaphasic plane. When these chromosomes are aligned at the equator, we call them metaphasic plate. This and one more thing is very, very important, that the chromosome study is done at this stage only. Okay. See here, this is the chromosomes gradually they are, this is here scattered, but they are coming to the middle. And here you see this is the metaphase. Here all the chromosomes, they have aligned at the central point, okay, equatorial plane, and they are attached to the spindle fiber. These are the centriole. Here you can see this white spot it is showing that these are the kinetochore. So here it is, sorry, kinetochore. And this one is a spindle fiber. Okay, so this period is very short period, and one thing we should remember that all the chromosome study, particularly chromosomal study, is done at the metaphase. Metaphase of mitosis, because at this stage the chromosomes are very short and thick, and each chromosome is with two chromatids. Say, one thing we have to remember, that the chromosomes where in the prophase each chromosome has two chromatids, in the metaphase also each chromosome has two chromatids. After this is the anaphase, and anaphase begins with the separation of this chromatid. Say there are two chromatids, sorry, two chromatids are there. These two chromatids will separate. When these two chromatids will separate, then start the anaphase. So here is a separation of sister chromatid. Sister chromatids, so we call the chromatids of the same chromosomes are sister chromatid. All right, sister chromatid. When it is separated, we can call them that the daughter chromosome. So here now, each chromatid is a chromosome. They are now treated as chromosome, but we call them their daughter. Okay, so now each chromosome is having one chromatid. See here, this this diagram, the chromosomes has separated and they have started moving to the opposite pole. Here it is separation of sister chromatids and their movement to opposite poles. Okay, so they are moving to the opposite poles. This is anaphase, and at anaphase each chromosome has one chromatid. Now next stage, this one is also is short. After that, gradually they will reach to the opposite pole, and when they reach to the opposite pole, anaphase, centromere of chromosome divide longitudinally, separating sister chromatids of each chromosome. Sister chromatids moves towards the opposite pole. These sister chromatids, we can say them as daughter chromosome. Okay, after that telophase, this daughter chromosome or the sister chromatids have reached to the opposite pole, and the telophase is having all the event opposite to the prophase. whatever has happened in the prophets, the same but opposite direction, this all those events will happen. In in the prophets, there was condensation of the chromosomal material. Here it is a decondensation of the chromosomal material. Okay, and so the gradually this identity of the chromosome is lost. So they become very long, thin thread. All right, and nuclear membrane start appearing, the nucleoli also appear, then Golgi complex, endoplasmic reticulum also appear in the cytoplasm. Okay, see here, this have reached to the opposite pole. This is telophase, and around them the nuclear membrane has started forming, and from here, from one nucleus, two nuclei within the cell has formed. Is it? Two nuclei has formed, but after that there is cytokinesis. Then after telophase, there is cytokinesis. Cytokinesis is the division of cell cytoplasm. Here is cytokinesis is the division of cell cytoplasm. All right, this is, and this thing takes place, this thing takes place in different way in plant and animal. In animal, it is called, the process is called cell furrowing. Cell furrowing, and in plant, it is called cell plate formation. Okay, this is the two method that is. See what happened here. This process of cytokinesis start when this cell is in the anaphase stage. From the anaphase it started. Okay, late anaphase. It has started. In the animal cell, there is no cell wall and the membrane is very much flexible. So what happened, that say there is a cell, this cell start a forming a furrow, a depression, and it gradually moves towards the center. Okay, so after that it has moved towards this, and ultimately two cells have formed, and in between the nuclear are there. This is called cell furrowing. A furrow is formed. But in case of plant cell, there is cell wall, and so the cell cannot make a furrow like this. So what happened in those cells, in plant cell, there is a deposition of a structure, cell wall material, that is known as cell plates. Okay, these actually represent the intermembrane, that the middle lamella. You know, middle lamella. Middle lamella is the structure that is present in between the two neighboring cells. So this small Cell wall materials are deposited along the equator and ultimately two cells are formed. So this is middle lamella material is deposited first, and on its both side cell wall is formed. So from the one cell, two cells are produced. Okay, one thing is there, one very important event it happened. Always the cell karyokinesis, the nuclear division is not followed by cytokinesis. If the cytokinesis is not followed, then what will happen? Within the same cell, there will be two nuclei. If again there is a division, from and further this cytokinesis does not take place, there will be more number of nuclei. This condition is known as the syncytium. In some organism, karyokinesis, that is division of the nucleus, is not followed by cytokinesis, as a result of which a multinucleate condition leading to the formation of syncytium is formed. This liquid endosperm of coconuts is the example. Liquid, that water, coconut water, is that is the liquid endosperm, and it is the that syncytium. What is the importance of mitosis? Mitosis is the general cell division. It's from one cell, one mother cell, we get two daughter cells. Though these daughter cells are identical in their genetic complements. Daughter cells are identical to the mother cell and also to each other. Okay, because their genetic makeup is not changed quantitatively and qualitatively. Number of chromosome remain the same, and also quality of the chromosome and the DNA material. It is the means of multiplication. Say when bacteria divide by mitosis or unicellular organism divide by mitosis. So from one cell to two cells. So this is the reproduction of those cells, cell reproduction or cell multiplication. So unicellular organism, this is the method of their reproduction or multiplying. But for the multicellular organism, it is the method that is the basis of growth and development. Itself, somewhere there is injury. Okay, injury is healed by making new cells. So this cell healing and cell repairing, all this is done by mitosis. Okay, mitosis help in the cell replacement also, that old cells are replaced by the new cells. And also there is sometime, have you seen in the wall lizards, etc., their their tail is lost, but a new tail is formed again. This is called regeneration of the organ. This regeneration of organ is by the mitotic cell division. Okay, so next we will have some question on this. Let's see here some of the question now based on this mitosis and cell cycle. First question: the replication of DNA is a prerequisite for a eukaryotic cell to undergo division. Is it? For the cell division to take place, their genetic material, that is the DNA, okay, this DNA should become double. DNA replication should takes place before the division takes place. So this DNA replication takes place when S phase, G1 phase, G2 phase, or M phase. In which phase this DNA replicates at in which phase?
space.
S phase. S is for the synthesis. So during the S phase, the DNA replication takes place. At the S phase also in animal cell, the centriole centriole also replicate. Okay, centriole also divide, replicate during the S phase. In plant cell, of course, the centriole are absent, so there is no question of this. This was asked in AFMC 2007. Now there is only NEET exam for all the medical. Says the plant cell can show mitotic division in diploid cell, haploid cell, polyploid cell, both 1 and 2. Which one? Plant cell, which cell undergoes mitosis? Which type of cell? Diploid cell divide by mitosis or haploid cell divide by mitosis? In case of plant, same. In animal, in animals, usually in general, the diploid cells divide by mitosis. All right. But in case of plant,
Both
it is both
haploid and diploid.
haploid as well as diploid. So you cannot answer this one, you cannot say this one. Best the answer is that both 1 and 2, that is diploid and haploid. So our answer will be number 4, both 1 and 2. Say this is polyploid. Do you understand polyploid? Haploid, the cell having only one set of chromosome, that is haploid. One set, one set of chromosome. Diploid, that is diploid. All right, you can pronounce this at diploid. Di means two, so two sets of chromosome. All right, and if like that, this haploid and diploids are the most common thing. Okay, but some of the organism, due to abnormality and other causes, what happen, they have three sets, four sets, or even more number of sets. When the number of sets of chromosome in a cell or in an organism is more than 2, then they are known as polyploid. So mitosis division can take place in polyploid cells also. So in case of plant, it is haploid, diploid, polyploid in all. But we have to select here from the option given, and the best option is number 4. Next question. A somatic cell, a somatic cell that has just completed S phase. S phase has been completed of its cell cycle as compared to gametes of the same species. This is a very tricky question. uh, as compared to gamete, the that cell That somatic cell which has completed S phase, that will have how many chromosome and what will be the amount of DNA? Twice the number of chromosome and twice the amount of DNA. Say, we should remember that gametes of an organism have haploid sets of chromosome. Gametes are haploid. Normal gametes are haploid, means one set of chromosome, and their somatic cell means a normal body cell. Here is the somatic cell and gametic cell. The somatic cells are diploid. Okay, remember this. Somatic cells are diploid, two sets of chromosome. So normally this gamete, one set of chromosome, one C of DNA. Amount of DNA will be 1C. Somatic cell, they are diploid, they will have 2C of 2C content of DNA. Now the cell has completed S phase, that is DNA replication has taken place. In that case, what will be the number of chromosome and the amount of DNA? Twice the number of chromosome and twice the amount of DNA. Here, same number of chromosome but twice the amount of DNA. In compared to the gametic cell, here twice the number of chromosome and four times the amount of DNA. Four times the number of chromosome and twice the amount of DNA. This was asked in 2015 CBSE AITMT. Very important question from the cell cycle. What is there? See, somatic cell, as I told, it is asking somatic cell. In the somatic cell, normal condition is two set of chromosome. All right. So in compared to gamete, it is two times, twice the number of chromosome. But as it has undergone S phase, in the S phase, the DNA synthesis takes place, and the DNA content become double. So this DNA has become from 2C to it has become 4C. So chromosome number will be double, but the DNA content will be 4 times. So twice the number of chromosome and 4 times the amount of DNA. So this will be the answer. Okay, this will be the answers. CBSE PMT. And next, another question, easy one. Do you have any doubt here in solving this question? Do you have any doubt?
No, sir.
No. Okay. Then next question one. During which phase of cell cycle amount of DNA in a cell remain at 4C level if the initial amount is denoted as 2C? In which stage it will be? Here it is G0, G1, G1 is only G2, G2M. In which one? This was also asked in the CBSE ITMT. What will be the answer? What is the answer?
Sir, only G2.
Yeah, it is. It will be only G2. Of course, I have put a star asterisk here. Hmm. It is say in the DNA synthesis 4C condition. 4C condition will take place due to this S phase. So in the S phase, it is in the S phase. But here S is also written and G1. G1 at the G1 phase, it is not. DNA synthesis does not take place. During the G1 phase, the cell will grow only. Is it? Young cell will grow. So it is not that. So this cannot be the answer. All right. So it is in the S phase, but this one we cannot. After the S phase, G1, S, and G2. Here takes place the replication. So G2 will have 4C. S will have 4C by the end of the S. It will be 4C. And G2, it will be 4C. And after that, where? After that, in the prophase also, chromosome number is 2n, but it is within one cell, there is 4C amount of DNA. Prophase, then metaphase. In the anaphase also, the cell will have the 4C condition. But when it is telophase, in the telophase, the cell will divide. Cytokinesis will take place and the cell divide into two. So here it will be in this, sorry, telophase and G1 phase, this will be again 2C condition. Up to here, it is 4C. This one also will be 4C, 4C, 4C. Here it is written G2 and M. M is the dividing phase, but not in the entire M, because telophase also is the part of the M, is it? So this cannot be. That's why we have to tick this one, this M phase. Okay. So today it is up to here. In the next class, we'll discuss we'll solve the